Hi everyone, I'm Wudi from Department of Neurology, Zhenwu Hospital, Beijing, China. I'm honored to be here to introduce our latest study entitled A Vertigo Network Derived from Human Brain Lesions and Brain Stimulation, which is now published on Brain Communications.
As we know, vertigo is one of the most common neurological complaints. Classically, conditions causing vertigo are linked to pathology involving the vestibular larynx and the subtentorial structures. In addition, studies in non-human primates and functional neuroimaging in humans have identified certain cortical regions as the vestibular cortex. But there are case reports of vertigo in association with highly heterogeneous cortical lesions, suggesting vertigo involves specific cortical regions or networks. This brings an important question. The cortex functional network of vertigo remains largely unknown until now. Neuroanatomy-based traditional lesional mapping were typically used to localize brain function. However, it has its own limitations, and it is hard to localize neurological syndromes that incorporate such network effects. A recently developed technique, termed lesion network mapping, can test whether different brain lesion locations causing a specific syndrome map to a common brain network. This method has successfully been used to identify brain networks associated with a series of lesion-induced neuropsychiatric symptoms. So, in the present study, we aim to evaluate whether vertical lesions are part of a common brain network. Then, to define stimulation network localization of vertigo based on this method by using stimulations elicited vertigo. And finally, to evaluate the correlation between lesion and stimulation network localization of vertigo. Here are the inclusion criteria and exclusion criteria of the study. And here's the procedure of the study. Here's the workflow for identifying cases of vertical lesions from the literature. After carefully searching, 23 lesions were included in the study. In particular, 14 of these lesions were separated from the traditional vestibular cortex. Next, we conducted the connectivity analysis in 9-MNI space using a large resting state functional MRI dataset. To test for specificity, unthresholded lesion network maps of all vertical lesions were compared with the unthresholded lesion network maps from control lesions causing other neurological syndromes. Then, by conjunction analysis, the peak lesion network overlap in bilateral ventral posterior insula was both sensitive and specific. The network was defined by positive connectivity to lesion-based hub in bilateral ventral posterior insula. We shall refer to this network as lesion-based vertical network. Further, we computed a data-driven, coordinate-based direct electrical stimulation vertical network. By comparison, stimulation sites eliciting vertigo were more to the lesion-based hub in bilateral ventral posterior insula compared to the stimulation sites not eliciting vertigo. We found that 86% of the stimulation sites eliciting vertigo fell within the lesion-based vertical network. We computed a data-driven stimulation-based vertical network. Both the sensitivity map and the specificity map derived from stimulation sites show similar topography to a lesion-based vertical network.
Finally, the human vertical network was depicted. The vertical network includes primary vestibular cortex, including PIC and OP2, and it also contains regions outside this area, including somatosensory cortex, higher level visual area, synchronous visual area, and the thalamus. Overall, we identified human vertical network based on brain lesions and brain stimulations, and we provide novel insights into the network organization of vertigo.
We would like to thank our founders, collaborators, and data providers who make this research possible. Thank you for your attention.